All right, great jobs. Now let's move on to RNA. We've done DNA. We understood that DNA is a double helix structure, and it's made up of nucleotides. The nucleotides of DNA are made up of your phosphate, your sugar, that's phosphate, sugar, and which sugar is it made up of? Deoxyribose sugar. And then it has nitrogenous bases, which are adenine, which forms a base pair with thymine, and then guanine, forming a base pair with cytosine, and then the pairs are basically paired via weak hydrogen bonds. So that is DNA. We looked at the role of DNA and so forth. Now let's come to RNA. What is RNA? RNA is known as ribonucleic acid. Ribonucleic acid. So what are we going to be looking at when we look at RNA? Just RNA. Let's summarize what we're going to be looking at. Right. First things first, we'll be looking at the location of RNA, and then we're going to look at the structure of RNA, and then we will look at the role of RNA. What role does it play in the cell? Great. And then lastly, we'll be comparing RNA and DNA. So basically that's what we're going to be doing in this lesson. It's going to be a short one because RNA is, I presume, is simple enough, right? Good. Now let's start. Let's look at RNA. What is RNA? We're going to start with the structure of RNA. The structure of RNA. RNA is a single, first and foremost, the structural difference between it and DNA is that it's a single strand. If you look at the picture that I represented, the both of them right now, is that you'll find that DNA has two strands bonded together, and then RNA is single-stranded. So it is not coiled, and it does not form a double helix. That is the structure of what? Of your RNA. Secondly, if you look at RNA differs from DNA in the fact that there is no thymine. Remember thymine, the nitrogenous base thymine? It has no thymine. And then, lastly, we're going to be looking at RNA from a sugar point of view. It's pentose sugar. The sugar that is made up of is actually not a deoxyribose, it's a ribose sugar. So those are the three structural differences that we're going to be looking at. If I was to be asked to draw one very quickly, I would simply go and say, here is a nucleotide, a single nucleotide for your RNA. This is made up of a ribose sugar, and then a phosphate. It has a phosphate just like DNA, but a ribose sugar. And then it has nitrogenous bases. But its most unique nitrogenous basis is uracil. Instead of thymine, it has uracil denoted by capital letter C. So that is just a typical nucleotide structure for RNA. So it's another form of RNA is another form of nucleic acid. Right, it has its own, it has almost the same monomers. They just differ on certain places. Right, good. So that is the structure of your RNA. So now let's move on. Let's look at the location of RNA. where do we find RNA? So I'm going to couple it with basically the types of RNA. Because there's three types of RNA. Yeah? So there's three types of RNA, and it depends where you find them in the cell. So, for example, the first one is known as messenger RNA. Messenger RNA, location GPS. It's found where? It is going to be found in the nucleus. It will be found in the nucleus. Now, what does messenger RNA do? Messenger RNA basically takes a genetic code, for example, for a protein, and then leaves the nucleus, goes into the cytoplasm where you find ribosomes, and then attaches to the ribosome. Yeah? Then we have what we call ribosomal RNA. Remember, there's a short word for it, is rRNA. So small letter R and then capital letter acronym for the RNA. And then this one will be small letter M and the acronym RNA. So it's mRNA. And then 3, it is the, if it's not messenger RNA, if it's not the ribosomal RNA, we have the transfer RNA. RNA, or the tRNA. Where do you find the tRNA? tRNA is found in the cytoplasm. And then where do you find a ribosomal RNA, which is obvious, in the ribosomes? Right? Good. So what is the function of the transfer RNA? Basically transfers your amino acids from the cytoplasm to the ribosome to manufacture what? To manufacture proteins. I had an image over here. Yes, this is a good image to represent, to show you that, but we'll get into detail when we do protein synthesis. Basically, your mRNA, your messenger RNA, is made from your nucleus. It's made in the nucleus, and then it leaves the nucleus and then goes to the ribosome to deliver the genetic code for the protein maybe your cells want to make. Right? Once the mRNA gets there, it attaches to the ribosome, and then the transfer RNAs, these are transfer RNAs. What do they transfer? They transfer amino acids to what? To the ribosomes, and then those amino acids are stacked together, they bond together, they form peptide bonds up until it's a whole protein. So that is that is your RNA. So RNA, when I say location, it depends on what type of RNA we are talking about, right? We cannot just say RNA is found in the cytoplasm. Which type of RNA is found in the cytoplasm, right? Great. Now, what is the role of RNA? Since there's 3 types of RNA, we've already touched on it. There's 3 types of RNA. Messenger RNA is to deliver the genetic code remember, a messenger RNA is made in the nucleus from DNA. It delivers the genetic code to code for specific proteins, for example. Then we have the transfer RNA. The transfer RNA transfers what? Amino acids from the cytoplasm to the ribosome to form peptide bonds and ultimately form protein. So basically, if I was to tell you the role of RNA in a nutshell, is for protein synthesis. It helps with protein synthesis. It helps with, remember, proteins are made in the ribosome, right? Good. So that is your RNA and the structure of RNA. So what is important to remember? The structural difference between RNA and DNA, and then the role of the 3 types of RNAs that we find: the messenger RNA, transfer RNA, and the last one being your transfer RNA. Are we together? Great. Now we can basically just summarize the differences and the similarities between the 2 uh, nucleic acids, right? Let's try that. Let's start with the similarities. I'll write them in red. So the similarities, they contain what? They contain a pentose sugar, right? And that pentose sugar alternates with a phosphate. What do we mean by this? Remember, you find a phosphate in both RNA and DNA, and then you find a sugar, doesn't matter which sugar, and then another phosphate, and then a sugar, then another phosphate, and then a sugar. So they alternate. It's phosphate, sugar, phosphate, sugar, phosphate, sugar. It doesn't matter on which side they have that in common. Ne? So we're not finicky on the type of sugar, just that we know that phosphate alternates with a sugar, a pentose sugar. Then the second one, they both have a nitrogen, they both have nitrogenous bases. They're nitrogenous bases. They have adenine, they have adenine, they have guanine, and they have cytosine. So they share these three nitrogenous bases in common, except for the fourth ones, basically. And they both play a role in protein synthesis. They both, so basically, remember when we say both of them play a role, remember DNA is the one that has the genetic code for a protein. And then how does it get it out into the ribosome? mRNA basically takes that genetic code into the ribosome, and then the protein will be synthesized with the help of other RNAs. So that is the similarities between the both of them. Basically, the comparison between the both of them. Now the differences. How are they different? We all know that DNA contains what type of sugar? Contains a deoxyribose sugar. And then RNA contains a ribose sugar. All right. RNA is a single strand, meaning you find it as a single strand. And then DNA basically has a double helix structure, and it's coil. Remember, it was discovered by Waston and Crick, yeah? Together with Rosalind Franklin and Maurice Wilkins. Then the last comparison, yeah? The last difference is that this one, RNA, instead of thymine, it has uracil in place of thymine. There's no, how I cram it, as I usually say, when we go to RNA, there will not be T for U. So there won't be T, there will be U, which is uracil. And then DNA, it has thymine instead of uracil. So that is the differences between the two. Great. And if my handwriting is bad, I have a couple of slides basically just to summarize what we just did. RNA is another type of nucleic acid, and we have three types: a messenger RNA formed in the nucleus and basically gets the genetic code, which they transfer to the ribosome from DNA in the nucleus. Ne? Good. Then ribosomal DNA found in the cytoplasm forms part of the ribosome, and then transfer RNA found in the cytoplasm picks up amino acid and takes them into the ribosome, where they bond together, forming peptide bonds to form that polypeptide, which we call a protein. And then another structural difference: it contains a ribose sugar instead of a deoxyribose sugar. it contains a phosphate, and it has 4 types of nitrogenous bases. However, it does not contain thymine, it has uracil. So it has all the other 3, adenine, cytosine, and guanine, but no thymine. Remember, there's no bonds when it comes to an RNA. Why? Because it doesn't bond to another nucleotide. Why? Because it's a single strand. Then we discuss the similarities because they both contain sugar alternating with a phosphate. If you look at the structure over here, there will be a phosphate and then a sugar, phosphate, then a sugar. Similarly, in DNA, it's the same thing at the backbone of your nucleic acid. Then contains nitrogenous bases, adenine, guanine, and cytosine. They only differ at thymine and uracil. They play a role in protein synthesis. Then their differences: contains ribo. DNA will contain your deoxyribonucleic acid, while RNA contains your ribose sugar. DNA has a double helix shape and it's square. It has 2 strands basically. Then this one is single-stranded. RNA is single-stranded, and then DNA contains nitrogenous base thymine, and then RNA contains nitrogenous base uracil in place of thymine. So that basically concludes my lesson on DNA and RNA.